Good afternoon, ladies and gentlemen, and dear colleagues. My name is Libor Kolesár, and I'm the Senior Field Application Scientist and Assay Product Manager at Omixon. And I would like to welcome you to our today's webinar, which is dedicated to HLA role in cancer immunoediting. I would like to welcome here our guest speaker, Dr. Milena Ivanova from Sofia, who will give us a presentation titled HLA Genotypes Shapes JAK2 polymorphism-driven oncogenesis. So welcome, Milena.
Thank you very much, Libor, for the kind introduction. First of all, I would like to thank the Omixon for inviting me to take part into this webinar and giving me the opportunity to present our recent work on HLA association in JAK2 V617F driven oncogenesis.
Yeah,
so,
welcome. Yep.
Okay.
But before we start, let me tell you a few words. So the agenda of our webinar is the following. After this introduction, Dr. Ivanova is going to present the results of her and her team's research. After the closing remarks, there will be plenty of time for questioning Dr. Ivanova over her presentation. You can find in the GoToMeeting toolbar a section called Questions, where you can place your question. So although we are not new to the HLA market, let me briefly introduce you our company and what we are doing. So Omixon is a global molecular diagnostic company with the headquarters in Europe, in Hungary, and manufacturing and operations in the US. Among our portfolio, you can find assays for pre-transplant HLA typing like holotype HLA, singleplex PCR-based typing assay, or OmniType, which is a multiplex PCR-based typing assay, as well as monotype HLA for typing HLA genes for HLA-associated diseases, monotype ABO and MIC-A for typing these genes. Among software products, we can offer to our customers two products, HLA Twin, which is the software for clinical use, and HLA Explore, which is a software for researchers. But without further delays, I would like to give the floor to our guest speaker, Milena Ivanova, who is a lab co-director and lab supervisor of the Histocompatibility and Immunogenetics Laboratory at the Department of Clinic of Immunology at the University Hospital Aleksandrovska in Sofia, Bulgaria. Although her everyday routine is HLA typing, she's also very active in the research field, focusing on the role of HLA in the immune editing in hematological malignancies and MIC-A research of certain types of oncological diseases. So the presentation is being recorded, so if you miss something, don't worry. Milena, the floor is yours.
Thank you once again, Libor. Myeloid malignancies are clonal diseases of hematopoietic stem cell or progenitor cells. They affect myeloid lineage proliferation and impair differentiation. These diseases result from genetic and epigenetic alterations that disturb the normal key processes such as self-renewal, proliferation, and differentiation, and therefore they share a common molecular feature. Generally, these diseases have poor prognosis, and currently allogeneic hematopoietic stem cell transplantation is the only curative options. Libor, would you please change the slides for me, because obviously there is a technical problem? Thank you. Recently, it has been postulated that chronic inflammation constitutes a trigger and driver of clonal evolution in myeloproliferative neoplasia, and inflammatory processes may constitute a major risk factor for malignant myeloproliferation or even leukemic transformation. So next, many studies over the last years demonstrated the important role of the immune system in resisting or eradicating formation and progression of neoplasms And avoiding immune destruction has been formulated as one of the hallmarks of cancer in a paper of Hannan and Weinberg. This paper provides a logical framework for understanding the remarkable diversity of neoplastic diseases. So next, our understanding, the next one, please. Our understanding how the immune system affects cancer development and progression has been one of the most challenging questions in immunology. The long-standing theory of immune surveillance proposes that cells and tissues are constantly monitored by ever-altered immune system, and that such immune surveillance is responsible for recognizing and eliminating the vast majority of cancer cells and nascent tumors. It has been shown that cancer undergo immune editing, and this is an extrinsic tumor suppressor mechanism that engages only after cellular transformation and failure of intrinsic tumor suppressor mechanism. In the most complex form, cancer immune editing consists of three sequential phases: elimination, equilibrium, and escape. In the elimination phase, innate and adaptive immunity work together to destroy developing tumors long before they become clinically apparent. Editing of tumor immunogenicity occurs in the equilibrium phase as a result of constant immune selection pressure over the genetically unstable tumor held in equilibrium. As a result, tumor cell variants may emerge that are no longer recognized by the adaptive immunity, that are insensitive to immune effector mechanisms, or that could induce an immunosuppressive state within the tumor microenvironments. These tumor cells may then enter the escape phase and cause clinically apparent disease. During the last years, several models have been proposed for immune editing of cancers, and these studies demonstrated the key role of HLA class 1 and more recently HLA class 2 molecules. And as you can see on the next slide, the structure of HLA molecules, which is very well known. We know that HLA class 1 molecules are expressed by almost all nucleated human cells on their surface, and these molecules present degraded cytosolic protein fragments in the form of 8 to 11 amino acid peptides for recognition by CD8 cytotoxic T cells. MHC class 1 is both polygenic, encoded by 3 loci, HLA A, B, and C, and highly polymorphic. This combination allows a diversity of peptides to be presented and also creates significant heterogeneity between individuals as to which antigens can be presented. Therefore, HLA diversity directly affects the repertoire of peptides. Cancer cells often retain their HLA class 1 function and may express protein products encoded by mutated genes, neoantigens, that select the cancer cells for elimination via the adaptive immune system. In a recent study, Marty et al. Provided strong evidence for immune editing mediated by HLA molecules. They evaluated the binding interactions between neoantigens and HLA proteins as a predictor for antigen presentation. By analysis of cancer genome atlas datasets, they found that individual patients are significantly more likely to have a mutation that gives rise to neoantigen with low HLA binding affinity, suggesting that highly presented mutations are selected against. Similarly, they show that highly recurrent oncogenic mutation generally have low HLA binding affinities across the population, which is consistent with the hypothesis that an oncogenic mutation is unlikely to be adaptive unless it is also immunologically silenced. Another mechanism that is commonly utilized by cancer cells for immune escape is downregulation of HLA class 1 expression. In another recent study, it has been shown that HLA antigen presentation affects tumor evolution, and this could be predicted by evaluating the effect of HLA copy number loss. In solid tumors, it has been shown that the frequency of loss of heterozygosity correlates with both an increase in neoantigens predicted to bind to the lost HLA alleles and with an increase in the overall number of subclonal mutations in patients with adenocarcinoma. And the authors of this paper proposed that the observed increase in neoantigen load is due to compromised MHC class I antigen presentation resulting from HLA loss of heterozygosity. Next, please. Unlike solid tumors, hematological malignancies are characterized with lower mutational burden, and therefore they are less likely to produce immunogenic neoepitopes. However, there are some evidences of the role of HLA in immune editing. Spontaneous CD8 T-cell immune responses have been documented in several studies of acute myeloid leukemia and chronic myeloid leukemia. Several studies also proposed the existence of protective HLA class I alleles for the development of CML, presumably because of their ability to present new antigens resulting from BCR-ABL1 fusion protein. MPN are a good model to exploit the hypothesis for the role of HLA in cancer immune editing. And now it is known that the key steps in MPN development include acquisition of MPN initiating mutation in a single hematopoietic stem cell. Then the mutant hematopoietic stem cell acquires a selective advantage over normal hematopoietic stem cells and also promotes myeloid differentiation, eventually leading to myeloproliferative phenotype. And the aim of our research project was to explore the hypothesis for possible role of HLA class I in immune editing that occurs in the early stages at the stage of mutated hematopoietic stem cells. Next one, please. In 95% of cases, a mutation that drive development of myeloproliferative neoplasias occurs in mutually exclusive manner in one of three genes, as you can see on the slides. These genes are JAK2, calreticulin, or MPL. The most common mutation, next one please, is JAK2 mutation V617F, which result in constitutive tyrosine phosphorylation and activation of Janus 2 kinase. Myeloproliferative neoplasia persist over a long period of time in patients, giving sufficient time for immune editing and indicating that JAK2 V617F mutation occurs in cells with long-term self-renewal capacity. And indeed, JAK2 V617F mutation is detectable in the early hematopoietic stem or progenitor cells that have been isolated by flow cytometry. And also in mouse model, JAK2 V617F has been shown to confirm a proliferative advantage on progenitor cells. Next one, please. The aim of our study, therefore, was to investigate the role of HLA immune editing in the development of JAK2 V617F-driven myeloproliferative neoplasia. And we hypothesized that if immune editing in JAK2 V617F positive MPN is taking place during the early stages of euchemogenesis, then there should exist protective HLA class I alleles able to present peptides derived from JAK2 V617F protein. Next one. We performed HLA typing by whole type HLA kits kindly provided by Omixon. And for data analysis, we used HLA twin software. Next one, our strategy for JAK2 V617F mutation detection included a Luminex-based approach that we developed and published previously. The sensitivity of method was 0.1%. Next one. In our study, we included 139 patients with myeloproliferative diseases positive for JAK2 V617F mutation, and they were compared to 622 healthy controls from the Bulgarian population. And as you can see from this slide, the control and patient group were biased in regard to age distribution, and that was taken into account during the data analysis. In our study, we applied population genetic approach to test the possible association. First, we ask, next one, please. First, we ask a question whether the diversity of the entire MHC class 1 genotype could be associated with the development of JAK2 V617-driven diseases. We tested whether homozygosity at any class 1 locus is associated with JAK2 V617F mutation. We did not find any association between the level of homozygosity at individual loci or in combination and the presence of mutation. However, we should keep in mind that as zygosity is assessed based on allelic designation heterozygosity at a given locus, it does not necessarily reflect significant diversity in the structure of peptide binding group and in the binding properties of the two HLA alleles in heterozygous states. Next, we analyzed the association with HLA divergence. HLA evolutionary divergence, or HET, for each MHC class 1 locus was calculated as described originally by Pierini et al. First, we aligned the protein sequences of exon 2 and 3 of the two alleles per given locus using Crystal W software. Then we calculated Grantham distance as a measure of HLA evolutionary divergence. Grantham distance is a global metric for evolutionary divergence of two peptides, which takes into account the differences in the amino acid composition of two aligned peptide sequences. Then, for each subject included in the study, we calculated the mean HLA evolutionary divergence as the mean of the heads of the three loci of the same subject. Next one, please. We estimated heads for every locus, as well as the mean HLA evolutionary divergence for all three loci in patient and controls. And it appeared that expectedly HLA-A and B loci had higher, significantly higher HLA evolutionary divergence compared to HLA-C locus. Notably, JAK2 V617F was not associated with any difference in head at any locus and mean head. This then we further assessed the theoretical ability of HLA class 1 genotype of any subject to present peptides derived from JAK2 V617F mutant protein. And as you can see on the next slide, we followed the procedure that was originally developed by Marty et al. To derive the theoretical best binding rank for each locus, as well as the patient best harmonic mean rank for each subject in the study. First, we generated the sequences of all possible octa, nona, deca, and decamers harboring JAK2 V617F mutation, and we identified 38 different peptides. Then this set of peptides was subjected to antigen binding prediction by each of the identified HLA class 1 alleles using NetMHC PAN4 server. The lowest binding rank obtained for each locus was assigned as best rank, and then we calculated patient harmonic mean best rank as the harmonic mean of best rank for all 6 alleles in each subject. The higher best rank is reflecting the worst theoretical binding property of the alleles at a given locus, whereas the higher patient harmonic best rank is reflecting the better antigen binding property of the entire HLA class 1 genotype. As you can see on the next slide, it appeared that a number of alleles could theoretically bind limited sets of nona and decamer mutant peptides. However, patient harmonic best rank did not differ between patients and healthy controls. Then, next, we performed a correlation analysis between the structural and functional diversity of HLA class 1 genotype represented by mean HLA evolutionary divergence and patient harmonic best rank. And as you can see on this slide, there is no significant correlation in patients and in control groups. Collectively, these data suggested that global MHC class 1 genotype and its diversity have little, if any, protective role for the development of JAK2 V617F driven myeloproliferative diseases. Our data are consistent with the report of Marty et al., showing that patient harmonic best rank has different effect in different types of tumors, and in acute myeloid leukemia, it has little, if no, effects. Next one, please. Then we tested the hypothesis for protective MHC class 1 alleles. That should be significantly less frequent in MPN patients, and that should be able to present peptides derived from new epitopes harboring JAK2 V617F mutant peptides. HLA association analysis was performed by fitting additive generalized linear model with the gender and age above 50 taken as covariates. As you can see on this slide, 3 HLA alleles were observed with relatively high frequency, more than 10% in both patients and control, and the highest variation in the allele frequencies was observed for HLA A0201 allele. On the next slide, you can see the distribution of HLA B alleles in patients and controls, and the highest differences were observed for B3501 and B4101. On the next slide, you can see the distribution of HLA C alleles with significant differences observed for HLA C 1203, 1502, and 1701. On the next slide, you can see the volcano plots of p-values versus odds ratio for HLA class 1 alleles. And 3 alleles were significantly less frequent in JAK2 V617F positive patients. These were HLA A0201, B3501, and HLA C1502. On the other hand, we found 4 other alleles to be significantly positively associated with the presence of JAK2 V617F mutation. These were B4403, B4101, C1203, and C1701. Then we analyzed B and 3 Wolfsay haplotypes, and the analysis were performed using haplostat package through implementation of imputed haplotype score calculation. P-values below 0.5 when considered significant. And as you can see on the next slide, B-Wolfsay haplotype analysis showed that the haplotype HLA A0201, B3501 was most significantly depleted in JAK2 V617F positive patients. To provide some mechanistic evidences, next we rechecked the predicted binding ranks for all 38 peptides that can bind to HLA class 1 molecules. And all peptide binding prediction were performed using NetMHC PAN4. Server and stability of peptide binding was performed using NetMHC Step 1 server. On the next slide, you can see that among those peptides, 1 nanomer, shown on the top of the graph in red, was predicted to be a strong binder for B3501 by the predefined criteria of the used NetMHC PAN4 server, binding rank above 2%. Furthermore, NetMHC Step 1 server predicted that among all peptides, this peptide was the most stable binder to B3501. Next slide, please. In vitro presentation of HLA class 1 antigens requires also efficient proteasomal degradation and efficient TAP-mediated transport to endoplasmic reticulum. Proteasomal cleavage prediction, and therefore we assessed proteasomal cleavage prediction by NetShop 3.1 server, and prediction of binding of peptide binding to transporters associated with antigen processing protein complex was performed by TAP-PRED server. Our results showed that our best binding peptides can be easily produced by proteasomal degradation from JAK2 V617F mutant protein, and this predicted best binder also is the best binder to TAP transporter proteins among all possible nanomer peptides harboring JAK2 V617F mutations. All these analyses suggest that indeed B3501 could be a candidate for protective allele for the development of JAK2 V617F driven MPN by the presentation of JAK2 V617F derived neoantigens. Then, as you can see on the next slide, we performed a molecular dynamic simulation of B3501 in complex with peptides. Actually, in these simulations, we used 2 peptides. Our peptide that is predicted to be best binder and predicted non-binder. And all these simulations were performed using NAMD, implemented through QuickMD interface. We used the initial crystal structure that was a complex of B3501 with a monomer deposited in the protein database, and then we mutated the initial binding peptide to the desired primary structure. So molecular model is seen on this slide, and as you can see, phenylalanine, which is actually the mutant amino acid, is shown here in green. Then on the next slide, we compared the root mean square deviation or RMSD values for the alpha carbon atoms for B3501 molecule, beta-2 microglobulin, predicted best binder, predicted non-binder. And as you can see from these slides, for B3501 molecule, beta-2 microglobulin, and predicted best binder, RMSD values remain stable during the 20 nanosecond stimulation, and the values were around 2 angstroms during the entire simulation. On the bottom of the slide, you can see a simulation in which we used a predicted non-binder. In this simulation, RMSD values for non-binder quickly reach 4 angstrom soon after the beginning of the simulation and remain around that value during the entire simulation. On the next slide, you can see the comparison of the distribution of RMSD values for each protein in the 3 simulation. And as you can see, RMSD values for predicted best binder were significantly lower compared to a non-binder or empty molecule. Collectively, these data suggest that the neoantigen that we identified in our study as a best binder fulfills the requirements for effective antigen processing and presentation by specific HLA B3501 molecule. Next slide, please. As our top hit protective allele, B3501, was not absolutely depleted among JAK2 V617F positive MPN patients, we reasoned that there might exist several other mechanisms that would be responsible for escape of transformed JAK2 V617F positive hematopoietic stem cell from HLA B3501 mediated immune editing. And the most reasonable explanation would be that on many occasions, pre-leukemic cells downregulate the expression of molecules involved in antigen processing and presentations by MHC class I. We therefore specifically investigated the expression of HLA class I molecules by CD34 positive cells in patients with MPN versus healthy controls. In our study, we used publicly available data sets and selected the genes listed to participate in the class I antigen presentation and processing by Kyoto Encyclopedia of Genes and Genomes. As you can see from these graphs, actually the previous one, please, a number of genes, including HLA class I, HLA A, B, TAP1, tapazine, were significantly downregulated in CD34 positive stem cells from JAK2 V617F positive patient compared to healthy controls. However, as you can see on the next slide, such downregulation, at least for HLA class I genes, HLA A, B, TAP1, and tapazine, was not observed on peripheral blood granulocytes from JAK2 V617F positive MPN patients. However, it remains unclear how many HLA molecules are expressed on the surface of malignant CD34 cells. As recent studies showed a broad range, also it is unclear how many HLA molecules needs to be expressed on specific neoantigens to be effectively recognized by T cells. Next slide, please. Besides these, B3501 might be an appealing candidate for immune editing in JAK2 V617F positive MPNs because it has some specific features. As it was recently shown by Genk et al., B3501 can be expressed on the cell surface as empty conformers and therefore can enhance the strength of immunological synapses between antigen-presenting cells and CD8 T cells. Another intrinsic feature of B3501 that is shared by another allele that was observed as a protective in our study, HLA C1502, is their relative independence of tapazine-mediated peptide loading, and therefore these alleles are less sensitive in terms of antigen presentation to low level of tapazine. The latter property might be of particular importance as many cancer types evade immune surveillance through downregulation of proteins involved in MHC class I presentations we already seen. These effects might be even more pronounced for some HLA alleles, including B3501, with intrinsically lower cell surface expression. And in conclusion, next slide, please. Our results showed that HLA class I genotype has no role in initiation of JAK2 V617F positive MPN. However, specific HLA class I alleles could be relevant for JAK2 V617F driven oncogenesis. For our population, we identified such allele, B3501. However, we need to validate these data in functional assays. B3501 allele was predicted to bind an anomar peptide derived from JAK2 V617F mutant protein. We also shown that JAK2 V617F positive stem cells might escape immune surveillance through downregulation of expression of HLA class 1 molecule. And therefore, presence of specific HLA class 1 alleles might be a predictive marker for response to certain immunotherapies upregulating HLA class 1 molecule expression. However, in our study, we also identified that specific HLA alleles and haplotypes could promote leukemogenesis, and we are currently looking into that. And I would like to finish by acknowledging the colleagues who were involved in this study, Velizar Shivarov, who performed the majority of simulation analysis, also hematologists from our hospital, Gilgana Svitkova, Evgeny Khadjiev, and another collaborator from Sofia Med Hospital, Angel Stoimanov. And I would like to acknowledge also people from our research group in the Department of Clinical Immunology, postdoc Svetlana Lukanov, PhD students, Bushra Khadra, and technologist Lydia Tsitsiaukova, Elena Tudorova, and Tanya Nedialkova. And last but not least, I would like to thank the Omixon for kindly provided reagents for HLA typing of part of the subjects included in this study. And the study was also partly supported by scientific grants. Thank you very much for your attention.
Thank you very much, Milena. This was an excellent presentation. So the presentation is now open for questions, for discussion, but before we get there, I would like to give you some time to think of your question. And I would like to mention two things. First, if you go to our website, omixon.com, go to the section Omixon Academy, where you can find the recording of our previous webinars. And this webinar will be also available there later on. And each of you will also receive a link with receive an email with the link with the recording of this webinar, and also with the link to the questionnaire. And the attendance on this webinar provides continued education credits accredited by the American Board of Histocompatibility and Immunogenetics. And you will find more information on how to request the ABHI credits in the follow-up email. And last but not least, feel free also to visit our YouTube channel, when you can find more videos related to our products and more. So thanks again, Milena, for your excellent presentation, and the audience for the attention.